My name is Joe Fox. I'm a professor of chemistry and biochemistry at the University of Delaware, where I also lead an NIH-funded Center of Biomedical Research Excellence. Research in my group centers on the development of new reactions for chemical biology. Our work is rooted in physical organic chemistry and organic synthesis, and we utilize the reactions we develop in a variety of applications, including live cell and in vivo imaging, drug discovery and drug delivery, radiochemistry, and biomaterials research. The nature of our work is highly multidisciplinary and involves collaborations with groups across the country and here at UD. It has been exciting to see how the chemical tools for bioorthogonal chemistry from our labs have become broadly adopted across academia and industry, and have recently advanced to the point of clinical trials in humans. We have partnered with local industry and Sigma Aldrich to make reagents developed in our labs available to the broader research community. We also have a rich collaboration with the pharmaceutical company Pfizer to develop new reactions and discovery tools that are needed in the pharmaceutical industry. Equally important is the science. My job as a faculty mentor offers me the opportunity to work with talented graduate student and postdoctoral researchers. Over the past 20 years at the University of Delaware, I've mentored more than 30 PhD students, 10 postdocs, and numerous undergraduates. I've had the opportunity to work with people from all over the globe, bringing a diversity of scientific and cultural perspectives to our research group. Former co-workers from my group have gone on to amazing scientific careers, both in industry and academia. 4 of my former co-workers are now faculty members themselves, and it has been incredible to see their careers and their own students develop. Now, I'll turn things over to some of my current students who can tell you a bit about themselves and about their research.
Hi, my name is Jessica. I'm from Scranton, Pennsylvania, and I graduated from Elizabethtown College with a degree in chemistry. My research in the Fox Lab focuses on synthesizing a diverse array of transcycle octenes using a new diastereoselective platform developed in our lab for applications in rapid bioorthogonal reactions. My work largely involves organic synthesis, photochemistry, and kinetic analysis. Outside of lab, I enjoy spending time with my dogs, picking fresh produce at Milburn Farms, and starting my mornings with HIIT workouts. At UD, I really love the collaborative atmosphere. We do a lot of collaborative interdisciplinary research within the Fox Lab, but we also collaborate with several other research labs on campus as well as with industry. You really get to be part of a team at UD, and as an organic chemist, it has been great to see the compounds I've made be utilized in so many different applications.
Hi, my name is Amanda Talon. I'm from New Jersey, and I graduated at St. Joseph's University with a BS in chemistry. The focus of my graduate research project is to catalytically turn on the tetrazine TCO ligation with a genetically encoded catalyst, allowing for spatially controlled imaging and drug delivery. My work involves a combination of organic synthesis, chemical biology, kinetic analysis, cell culture, and microscopy. One thing I really enjoy about UD is the collaborative environment among students in different labs, as well as the collaboration among different professors. This collaborative environment becomes really helpful when you've reached a bottleneck in your research and you need to consult with someone that may have different skills than you. Also, being in Joe Fox's lab, Joe has connections to industry, so it's nice that we get to collaborate with researchers in an industry setting. Outside of lab, I enjoy running, biking, playing with my dog, and recently I've started to play chess.
Hello, my name is Ashlyn. I'm a second-year organic student in the chemistry department. I graduated from Moravian College in 2019 with a Bachelor's of Science in Chemistry, and the focus of my graduate research project is on developing minimalist tetrazine carbohydrate probes to utilize the tetrazine TCO ligation to better understand cell biology. I'm also working on utilizing catalytic activation of the tetrazine TCO ligation to construct macromolecule structures and polymers. My work involves mainly organic chemistry, chemical biology, material science, and photochemistry, as well as some kinetic experiments. One of my favorite features of our department at UD is the NMR core facility. We have a large facility which makes attaining NMR data quick and easy. The NMR facility also has a great staff with Dr. Steve Bai and Dr. Caitlin Quinn, who are readily available to answer questions and help with data interpretation and experiment design. Outside of the lab, I enjoy reading and exploring new work. Some of my favorite places to get food near campus are Klondike Kate's and Bing's Bakery.
Hi, my name is Andrew Jemis, but people call me AJ. I am from New Jersey and graduated from Rider University with a degree in biochemistry. The focus of my graduate research project is to use photocatalysis for spatiotemporal control of bioorthogonal chemistry. Essentially, I use a reduced version of tetrazine and can oxidize it on demand using light and photocatalyst, allowing for an on-demand restoral of its reactivity. My work is a mixture of biochemistry, tissue culturing, microscopy, and photochemistry. Outside of the lab, I enjoy playing video games, weightlifting, music, and role-playing games.
My name is Olivia George. I'm from Texas, and I graduated from Texas A&M University with a BS in Biomedical Engineering and a BA in Mathematics. At UD, I'm involved in a collaboration between the Materials Science and Engineering Department and the Chemistry Department. The focus of my graduate research has been utilizing the tetrazine ligation reaction to pattern biomaterials for wound healing, drug delivery, and tissue engineering applications. In one of my projects, I utilized the rapid kinetics of tetrazine ligation to pull hydrogel microfibers in an interfacial polymerization platform. Tuning the backbone and structure of tetrazine and TCO molecules in the two phases has yielded fibers with interesting biological characteristics, including enzymatic sensitivity, hydrolytic degradability, cell adhesion, and enhanced mechanical modulus. My work involves a combination of polymer and small molecule organic synthesis, biomaterials, fabrication and characterization, microscopy, and tissue engineering. I love the outstanding facilities at UD and the collaborative atmosphere in the Fox Lab. Outside of lab, I enjoy reading, photography, outdoor activities, and baking, sometimes with the help of my 1-year-old standard poodle, Penelope.
Hi, my name is Jules Rosenberger. I'm from New Jersey and graduated from Albany College of Pharmacy with a degree in chemistry. The focus of my graduate research project is to use light to activate the release of biologically relevant molecules. One of my projects focuses on the use of light and photocatalysts to target drug release from inactive precursors. My other project involves using photocatalysis to activate electrophiles for identifying protein binding partners or protein drug targets. My work involves a combination of organic chemistry, chemical biology, and cell culture. Outside of lab, I enjoy playing soccer in a local rec league, going to breweries in the area, and taking trips into Philly for brunch or Philly Sixers or Flyers games. I love the collaborative environment at the University of Delaware, not only within our department, but also between departments on campus and with groups in the industry.
Hi, my name is Chuan. I'm from China and graduated from Colorado State University with a BS degree in chemistry. One of the focus of my graduate research project is to develop a cytocompatible photocatalyst that oxidized dihydrotetrazine to tetrazine. This oxidation could be utilized to in vivo activate the Diels-Alder reaction between tetrazines and transcyclooctanes, which is a very rapid bioorthogonal chemical reaction that has a potential application to chemical biology, radiochemistry, and material science. My work involves a combination of organic synthesis, photochemistry, physical organic chemistry, and kinetic analysis. A great thing about doing science here at UD is the core facilities are very supportive. They are very responsive and would able to help you with the highly customized experiment. Outside of lab, I enjoy spending time with my family and tasting the food that we made for each other.
Hi, my name is Sam. I'm currently a 6th year in Dr. Fox's lab at UD. I originally got my BS in chemistry from Trinity College in Connecticut. I have 2 projects that I focus on in this lab. One is using transcyclooctanes as sulfenic acid probes. So this would be a small molecule probe for oxidized cysteine, with the end goal being for chemical proteomics. And the second project is developing new techniques for bioconjugation methods. And so I think likewise, like a lot of my colleagues, these projects really focus on organic synthesis, biochemistry, chemical proteomics, and kind of the interface with all those.
Hi, my name is He Zhang. I'm from China and I graduated from Nankai University with a degree in chemistry. The focus of my graduate research project is to develop biocompatible materials based on tetrazine ligation our group has developed. My work focuses on the construction pattern materials and the microfibers that can serve as a platform for tissue regeneration. I'm also working on taking advantage of the photocatalytic bioorthogonal chemistry developed in our group to create injectable therapeutic materials that can be photocured as hydrogel in vivo. My work involves a combination of organic synthesis, photochemistry, and material science. Outside of the lab, I enjoy hiking, road trip, and visiting microbreweries. A great thing about doing science here at UD is that you will be able to get the chance to develop your project by collaborating with great people from different departments and even from other universities. I have had the chance to collaborate with the bioimaging group of Ziboli at University of North Carolina, and together we successfully were able to take a new reaction we developed and apply it to create potentially therapeutic materials in live animal studies. It is so exciting to see how those fundamental science from our group has a real biomedical applications.
Hi, my name is Parmesh Ramraj. I'm from India and I graduated from the University of Minnesota Twin Cities with a degree in chemistry. My graduate research project focuses on the synthesis of microfibers using tetrazine tricyclooctane ligation and the post-synthetic modification of these fibers with biologically relevant proteins for use as scaffolds in tissue engineering applications. My work involves a combination of organic synthesis, biochemistry, photochemistry, and material science. Outside of lab, I enjoy reading and watching movies.
Hi, I'm Jess, and I'm a fifth year student in the Fox group, and I got my chemistry degree from Williams College. My current project is the development of a true carbene dialkyl diazarine. The goal is to have a diazarine that can do CH insertion. And so my project is the interface of chemical biology, organic synthesis, and also photochemistry. Outside of working in lab, I enjoy powerlifting and also being out in nature. My favorite part of UD scientifically is the industry collaborations. So I've collaborated with Pfizer in my last two projects, and that's been great scientifically. And also it's opened a lot of doors career-wise as well. So that definitely is a great advantage of doing science here at UD.
Hi, my name is Sarah. I am from Chester County, Pennsylvania, and I graduated from Washington and Jefferson College with a degree in chemistry. The focus of my current research project is to find an efficient way to screen fluorescent dyes for use as photocatalysts to activate chemical reactions in biological settings. This is particularly relevant for catalytic turn-on of the tetrazine ligation, which enables real-time bioorthogonal labeling in live cells and in the in vivo environment. My work involves a combination of organic synthesis, photochemistry, and kinetic analysis. Outside of lab, I enjoy exercising and being the best cat/dog mom to my 4 fur babies. Two great things about doing science here at UD are the core facilities and instrumentation.